For those of you that did not attend lecture, too bad. I just remembered to record the lecture. Okay, it's just 10 minutes through. So, SDSPAGE electrophoresis or polyacrylamide gel electrophoresis. Those of you that were in food analysis might remember a little bit of the principle, but just an overview. SDS is short for sodium didosyl sulfate, and it's a reagent you mix with your protein solution so that you impart the total net negative charge to the protein, denature it, and break all non-covalent interactions. So basically, you will have a gel embedded within the cassette, which is a plastic compartment, and you will load your samples in each of these wells. So you would load either 10, 15 microliter, depending on the size of your cassette, and then you will, since all of them will have the same net negative charge, they will all migrate downward in your gel once the voltage is applied, and then the higher molecular weight will migrate slower, so they will remain at the top of the gel, and the lower molecular weight will migrate more downwards, and the size of the pores are dependent on the percentage of the acrylamide in your gel. So the higher percent of acrylamide, the smaller the pore size. And sometimes you can have a gradient. That means you can have different concentration of acrylamide throughout your gel, an increasing concentration of acrylamide from top to bottom. So once you run your gel, after a certain amount of time, you stop the run, and then you break the cassette open, and then you remove the gel, which you cannot see any protein bands yet. So you need to take the gel out gently without breaking it, and then use Comasi blue stain. Comasi blue is a dye that will bind to the protein, and then when you destain, the whole gel will, the blue color will be dissolved out, and only the protein will be stained blue, and then you can see your different proteins that have been stained. So you scan or you take a picture of your gel, and then you can see the distribution of your proteins. So you always run a molecular weight standard, so you can have an estimate. It's a qualitative measure, so you will have an estimate of the molecular weight distribution of your protein. In this example here, in this lane you have soy protein isolate. So soy protein is a mixture of different proteins. So it's not only one type of protein, so you'll see that there are different bands in that soy protein isolate. And then oftentimes we run a standard of this protein to determine, to identify the protein in our sample. And in this example here, what we did is we tried to identify the specificity of a certain enzyme when hydrolyzing the protein. So here we used papain enzyme to hydrolyze. We noticed that papain was selective for beta-conglycinin bands, and then pepsin enzyme in here was selective for glycinin bands, because the glycinin bands disappeared and more smaller peptides were formed. And then in the example of papain, the beta-conglycinin bands disappeared and peptides were formed. So by doing SDSPAGE, we were able to see the impact of hydrolysis and which proteins were hydrolyzed. So you will be running SDSPAGE gel, but also you're going to run into a sort of a problem with the casein, right? So the casein proteins, they migrate differently on the gel. So if you run casein on SDSPAGE, they will not migrate based on their molecular weight. They will migrate based on their size. So how are you going to tell me, what are you talking about? It's the difference between size and molecular weight. So the caseins are open structure protein. Whey protein, for example, is closed globular structure protein. So you will notice that the casein will not follow a standard movement in the gel because of that, because they're open structure, so they behave as if they're large proteins, but they're not really in molecular weight terms. They are large in volume, in diameter, because of the fact that they are open structure. So whenever people want to run casein proteins on a gel, they usually do urea gel. Urea gel is based on, separation is based on charge to molecular weight ratio rather than just molecular weight. But you won't be doing that in the lab. You'll just be doing SDSPAGE. But keep that in mind for you to, when you want to discuss this in your lab reports. They do have to discuss this in the lab. Okay. So, running the gel under reducing agent, under reducing condition is important sometimes. For those of you that will be running soy protein, I say, are they going to do reducing agent for soy?
I thought so.
Mm-hmm.
I have to see if it works with the timing.
Okay. So some proteins in nature are very large. They form quaternary structure. If you remember from your protein lectures in food chemistry, quaternary structure means they form, they associate via hydrophobic and hydrogen bonding and sometimes covalent interactions, disulfide linkages. So if you run them on the gel, you won't be able to see the separate protein subunits. It will be too large to see on the gel. So sometimes we have to run under reducing conditions. That means you mix your protein solution with a reducing agent. Either beta-mercaptolic ethanol is the common one, DTT is another one. But what happens is when you put the reducing agent, your disulfide linkages will be broken, and the large polymers will be separated into monomers, and then you can see them on the gel. And this is what I mean is that your soy protein forms a large quaternary structure, about 300 kilodalton in size, and they are stabilized, these red lines, they are stabilized by disulfide linkages. So they will, if you run them on the gel without reducing agent, this is how they look. They're going to look smeared and larger molecular weight. But when you apply reducing agent, then you can see the different banding of your proteins, the different protein monomers. So that's why you will, I think, use reducing agent for the soy protein sample. Okay, so the another structural characterization that is important for proteins is surface properties. You won't be doing this in the lab, so I'll just mention it briefly. So surface hydrophobicity is very important. It has a direct bearing on solubility and also emulsification and foaming properties. So, and surface hydrophobicity is impacted also by denaturation. So native protein might have lower surface hydrophobicity. Once it unfolds, its surface hydrophobicity will increase. Because when you unfold the protein, it will expose the hydrophobic residues in the interior, and when you measure surface hydrophobicity, you'll see that you've gotten a higher value. So if you want to determine, it's an indirect method for determining denaturation. So when you want to determine if the protein has been denatured, you compare it to a native protein that you know has not been denatured, and you measure surface hydrophobicity. And if it's higher, that means the protein has denatured. Surface charge. Surface charge, actually you're measuring the zeta potential, which is basically a surface charge. You can measure that by a chemical assay. You can measure it as well by an instrument. Here you're just measuring the net charge on the surface, and this also has a direct bearing on solubility and other functionalities as well. Free sulfhydryl concentration, also it's an indication of protein unfolding. Sulfhydryl groups in some proteins like whey protein can be hidden within the interior moiety of the protein, and then when the protein unfolds and you measure free sulfhydryl group, it will increase again. And then the amount of free sulfhydryl group that the protein has will give you an idea about, or an indication about, the susceptibility of polymerization. The higher amount of sulfhydryl group you have, the more prone the protein to polymerize via disulfide linkages and form polymers that are irreversible. Okay? You will be doing this in the lab, looking at amine groups or free amine groups. You'll be looking at the extent of amine blockage due to glycation of the protein or conjugation of the protein with a sugar or carbohydrate moiety. So via Maillard reaction. So if the Maillard reaction is occurring, it's a reaction between reducing sugar and amine group. So the more the reaction is occurred, that means more amine groups got blocked. So that's one method to determine extent of glycation or extent of Maillard reaction by measuring the amount of free amine. So you will be doing that. And also you won't be measuring, are they going to measure degree of hydrolysis?
Probably not.
Okay. So another application for measuring free amine using the octaldehyde reagent, OPA, is to determine degree of hydrolysis. You know that when you break down peptide bond, you release primary amines. So the more peptides are broken, the more free amines are released. So this is an indication of the degree of hydrolysis or the extent of hydrolysis. So if you want to determine how much hydrolysis has occurred due to hydrolyzing a particular protein with a particular enzyme, then you can use an OPA method to determine how much hydrolysis has occurred. So it can be used for two purposes, either blockage of primary amine or release of primary amine. Instrument-based analytical tools, differential scanning calorimetry. You will be doing that in the lab.
That's the plan.
That's the plan. So this instrument allows you to determine the onset of denaturation or the temperature at which the protein denatures, and it will also allow you to determine the extent of denaturation. Sometimes denaturation is partial, sometimes denaturation is complete. So the extent of denaturation is determined by looking at enthalpy. So DSC, here's a diagram for a DSC, which you will plot something similar to that. So if you look here, this is the temperature increase, and this is the heat flow on the y-axis. So basically you put your sample in a disk, a small disk, and you put it in the instrument, and the instrument applies heat, and then you monitor change in enthalpy as the temperature increases. So what this peak is called endothermic peak. So as you apply temperature, as you apply heat, at some point the protein is going to absorb this heat and break bonds and unfold. Okay? So that absorption of heat and breaking the bond is a change in enthalpy, basically. A change in the contained heat enthalpy. You've covered that in food chemistry, right?
That would have been Professor Labuza's section.
Okay. Do you know what enthalpy is? What is enthalpy?
If I remember correctly, it's the no specific heat.
Okay, anybody? What's entropy?
Entropy?
There's enthalpy and there's
Okay.
entropy. Yeah, Jesse.
Enthalpy, using energy, which creates heat, and entropy is absorbing energy. Okay.
I think you have them a little bit mixed up. Chris,
Are you talking about
entropy?
entropy too? Isn't the amount of disorder?
Yes. So entropy is the chaos. A lot of free energy, there's a lot of chaos. Enthalpy is the containment of this chaos. So you want to think about when the protein is formed in the cell, you form the primary structure of the protein, and then there's a lot of free energy. So there's a lot of movement. The protein doesn't want to do that. It wants to get to the most stable form to stop the movement, to contain the free energy into a contained energy. So bonds starts to form. Hydrogen bonding, electrostatic interactions, van der Waals, hydrophobic, covalent even. And then the more bonds are formed, the less entropy, because no more chaos. You reach a stable state. So when you're denaturing the protein, what's happening is that you're applying energy to break bonds. And once bonds are broken, there's a change in enthalpy, and then the protein unfolds. Okay? Yeah.
There's the SPI commercial line on the bottom. What is that?
Yeah, I was gonna get to that. Yeah, what is that? Yeah, what is that? Good. You noticed something. SPI commercials versus SPI produced. So this is produced in the lab. This is commercial. When you don't see any endothermic peaks, what does that mean?
Does that mean it's already denatured?
Yes, thank you. You don't see anything. It's already denatured. Whereas the one we prepared in the lab has 2 endothermic peaks for the 2 main proteins, glycine and beta-conglycine. So this you can see the temperature of denaturation, at which temperature you see an endothermic peak for one of the proteins and another one. And then you have also the enthalpy that caused the amount of change in enthalpy that resulted in the denaturation. And here we isolated glycine, which is this component from SPI, and we isolated beta-conglycine, and we compared the denaturation and enthalpy of the isolated fractions to the mixture of the 2 fractions following a different procedure. Yeah.
So when it's slowly dipping like that, is that just structural breakdown, or is it?
This?
No, the commercial line.
Oh no, it's just the baseline.
Okay.
Yes, I say nothing. No. So for the SPI commercial, there were no endothermic peaks. That means the protein has been fully denatured prior to our test. So other than DSC, how can we determine denaturation? Can you think of other ways? Okay. Yeah. Yes. Is it on your slides? UV light. So why? Well, UV light is correct, but your explanation is incorrect.
Don't peptide bonds absorb light at different wavelengths?
No. So peptide bond absorb light around 214 to 220, but proteins absorb light at 280 because of which components?
Amine group.
Huh?
Amine group.
Amine groups?
Yeah.
No.
That aromatic.
Yeah.
Amine structure?
What?
Amine structure?
Yes.
Yeah.
So which amino acids are these?
This tryptophan.
Tryptophan, phenylalanine, one more, the main one,
Tyrosine?
tyrosine. Thank you. So they do remember something from food chemistry.
Something.
And these groups are hydrophobic, right? And in general, hydrophobic groups hide away from water. So hydrophobic groups are within the protein tertiary structure. They're hidden inside. So once the protein unfolds, they will be exposed. Okay? So that means, okay, let me rephrase. So they are hidden, but some of them might be on the outside. That's why you always will detect UV absorption and fluorescence. But when the protein unfolds, more of them will be exposed, so more absorption will be detected, higher absorption. Okay, what happens to solubility when protein is denatured? Decreases. So measuring solubility of the protein is an indirect measure for denaturation. What happened to the digestibility of the protein? Yeah.
Decreases.
Okay. Under certain circumstance, it will increase to a certain extent. When your protein unfolds, it will become more accessible for enzymes. But if it polymerizes beyond denaturation, digestibility decreases. Okay? Polymerization causes reduction in digestibility. So you can measure digestibility and see what happens. If it changes from the native state, how it changes. Does it increase? That means it's unfolded. Did it decrease? That means it not just denatured, it polymerized. Okay. Electrophoresis. We just talked about it, but look here. So here we ran on a gel whey protein, non-heated. You have two main monomers, beta-lactoglobin and alpha-lactalbumin. And then we heated it at 75 degrees for 10 minutes intervals, from 10 minutes to 60 minutes. So see what happens. So when it was heated for 10 minutes, these monomers became fainter, and then you start seeing a dark band at the top. A polymer is formed. And then heat another 10 minutes more, darker here, fainter here. At 60 minutes of heating, you can see it got much darker at the top here in terms of polymer, and you lost almost all monomers. So SDSPAGE can give us an indication of polymerization. Polymerization post-denaturation. So other instrument methods for denaturation could be Raman and FTIR. Here you look at structural change by looking at IR absorption, or Raman, which is a diffraction of the light, or dispersion of the light. So here, for example, you have the whey protein isolate non-heated and heated. So you can look at different structural change, each band, that absorption of each well, see, I'm losing my focus. Each wave band, you see a different peak, and this would be a different bond within the protein. So different vibration of different molecular bonds within the protein. So you can see that if a molecular bond change, that would be due to change in molecular structure. So that's no heat and heat. So you can see that there's a difference here at pH 3.4, pH 4.5, 7, and 5.5. So you can detect major changes, and when you interpret the spectra, you can tell which changes actually occurred in that protein structure. So here is a glycated protein, and the glycation caused rigidity, prevented denaturation. So we can see that no matter what the pH was, no heat or heat, there was no structural change occurred. That tells us that the protein did not denature even when we heated, because the molecular structure remained the same, remained rigid.
So can this be a real-time scanning thing?
Real-time scanning?
Yeah, so like, if you gradually increase the temperature, do you monitor which boundaries first and so like a kinetic thing?
I don't know with Raman if you can do that. No, I'm not sure that you can do that with time, because with you have to prepare the sample in a certain way, so it's not a continuous. So you prepare your sample and you fix it on a plate with silver dendrite so that the silver dendrite capture the protein, and then you look at the structure after that. So it cannot be continuous. No. So you can't do mapping if that's what you're asking for.
Yeah, that's
No. Mm-mm. Okay, activity here, if the protein is an enzyme, and if you want to determine if the enzyme was denatured, you can do an enzyme activity assay. And if the enzyme lost its activity, that means it's denatured. Oh, this is another DSC showing the endothermic peak again. That's, I remember now why I have this here. So here, see how I told you this has no impact, heating had no impact on this protein is being glycated, and this means that the heating did not cause denaturation. So when we did DSC, the glycated protein actually had 15 degrees higher denaturation temperature than the regular. So the denaturation temperature was increased upon glycation. So multiple tests can be done to actually try to understand what happened to the structure and why. So you will be doing solubility measurements also in the lab, right? Yeah, okay. So determine how much the protein is soluble. The way you do that is you prepare a protein solution, either in a buffer at a fixed pH, or you adjust the water to a certain pH. And sometimes you would add salt to determine the impact of salt on the solubility of the protein. So you mix the protein in solution, and then you apply centrifugal force, and you take the supernatant and you measure the protein content in the supernatant, and you compare it to the original protein you put in solution. And by doing that, you would determine the percentage of the protein that was soluble. Because if your protein is not soluble, it will precipitate. So here is actually, instead of protein percent, it's protein solubility percent, it should say. So when you have whey protein isolate, non-heated, it has about 86% solubility. This is pretty low because this was done using colorimetric assay. But you'll notice that if your sample is whey protein isolate and you measure solubility using DUMAS, you'll notice that solubility of whey protein isolate will be really high, will be about over 90%, 95 to 100% soluble. That means all of the protein that you put in solution remained in the supernatant, was not precipitated. Okay? So if you prepare 5% protein solution, you'll find all of your proteins in the supernatant. That means it's 100% soluble. So like I said, the measurement of the protein in the supernatant would be either in using Keldal or DUMAS. I think in the lab you're going to use DUMAS to do that. Or colorimetric method. If you remember from food analysis, there are a bunch of them. One of them most common that we use in our lab is the BCA, bisynchronic acid assay. And it's usually a kit that you purchase and you need, if you use colorimetric assay, you really need a standard to construct a standard curve. So oftentimes it's BSA, which is bovine serum albumin, will be your standard. So you construct a standard curve, measure absorption, and then convert to concentration of the protein based on your standard curve. So here's an experiment we did in our lab a while ago to look at the effect of pH and temperature on protein solubility. And then here you have whey protein isolate, and there's the glycated protein that I told you about earlier. And we looked at solubility of whey protein non-heated. Whey protein is very soluble, so it remains soluble at all pHs, 7, 3.4, and close to the isoelectric point. It remains soluble 85 to 88% at isoelectric point. But when you heat it, it will come off solution. It will lose solubility at less than 5% soluble here. In this case, 2%. At 5.5, it is 5.8% soluble. At the isoelectric point, it's lost its solubility. The reason is that the whey protein is a globular protein that is very hydrophilic on the surface with only one hydrophobic pouch. So it's very hydrophilic, it interacts very well with water. Okay? So when it is in native form, not heated, even when the isoelectric point, when the pH is at its isoelectric point, there's not enough hydrophobic groups on the surface to attract the protein to each other and form polymers, so it will stay soluble. But when the protein denatures, there's a lot of hydrophobic groups exposed now. When the pH is at the PI, then there won't be repulsion, and there's a lot of hydrophobic groups exposed. The protein is going to come together and aggregate. Okay? So looking at the glycated protein, even at pH 4.5 and 5.5, it remained very soluble at the isoelectric point. This solubility is explained by the rigidity in the structure I showed you earlier. So I thought I had a different slide here. But see the structure, it did not denature upon heating. So it remained constant structure at 4.5 and 5.5. So looking at the structure and understanding the denaturation state would explain why the solubility remained high at 4.5 and 5.5. So the point I'm trying to make is that linking structure to function. Because structure did not change, it remained rigid, it remained non-denatured, the solubility was not impacted. Okay? Structure to function. Okay. What's the difference between water binding capacity and water holding capacity?
water binding capacity is the amount of moist water that a protein can absorb, and when it's being denatured or processed, water holding is the amount of moisture that it contains after processing over a period of time. How much moisture it's capable of holding after being processed.
Close. Josie.
Is water binding have more to do with the molecular interaction? Water with the product and water holding is more like synergy to me, just holding it like a gel structure.
Close. Oh, yes. She said gel structure. Yes. Water binding is the actual water interacting with the protein via molecular interaction, basically. So the protein and water interaction, this is the bound water that is not chemically available. It's strongly bound to polar groups on the protein. Whereas water holding is the amount of water that is physically entrapped within a three-dimensional structure of a protein. So a gel, how much water a gel can hold, how much water a beef patty can hold, okay? Are they going to measure water holding?
Yes.
Okay, so you're going to measure water holding capacity in the lab. Are they going to measure it by centrifugal force or by inverting the tube?
By inverting the tube.
Okay, so in order to measure water holding capacity, so you will form a gel at a certain protein concentration, and then you would invert the tube after you form the gel, usually thermally induced gel. You would heat until the protein denature and polymerize and form a gel matrix. Then you invert the tube and measure the amount of moisture, or measure before and after inverting, so determine how much moisture was lost and how much moisture was retained, and you do the calculation to determine water holding capacity. If you want to measure it in a food matrix, this is an example. Here you have a beef patty, and after cooking you've lost fat and water, and here's this beef patty that has been fortified with whey protein isolate to improve its water holding capacity and fat binding capacity. So if we want to determine the impact of an added ingredient, so what you can do, oh, I don't have that. I thought I had this. Usually what you would do is you get the meat and you put it in a cube. So you put your meat, after you measure fat and moisture content of the meat, then you heat it, and after that you let it sit overnight. Then you will have fat, water, and your meat. You measure the volume of fat and the volume of water. You convert using the density to mass, and you calculate based on the original fat and water content of the beef, and then you can determine water holding and fat binding. Okay. You will measure gelation by using using a texture analyzer to determine the amount of force needed to break the gel. And you can also, which you're not going to do, measure shear stress and shear strain and determine whether the gel is brittle, tough, rubbery, or mushy. You're not going to do that. You're just going to measure force needed to cut the gel, basically. Gel formation is affected by several factors. Do you know what environmental factors impact the gel formation? Yeah, that would be the last question I asked before I let you go. I got carried away.
well, instead of going with the safe answer, I was curious, the presence of calcium salts, does that play a role?
The presence of calcium, yes, will enhance gelation. Presence of salt, protein composition, yes, because you want a good balance between hydrophilic and hydrophobic. pH, pH, the temperature.
I understand.
Huh?
I will work on say.
Okay, so pH. If the pH is too high or too low, would the gel form? No, a lot of repulsion. If the pH is at the isoelectric point, you'll have a very gritty gel because the protein will aggregate rather than form a nice gel matrix. Temperature, you need to reach the denaturation temperature in order for the gel to form. What else did you guys say? And heating rate, you cannot heat you cannot boil the protein, for example, keep it boiling. What happens is the protein is going to denature fast and polymerize and aggregate rather than forming a nice three-dimensional gel. Okay, I was very slow today, so we will have to rush next time. Hello.
Do you feel better now? I feel like, did you see the appointment I'm with you on the calendar?
No. Hold on, let me.
Okay, so